Hi everyone. Today we're diving into the high-tech world of genomics biomarkers. These tiny molecular clues are helping us unlock which treatments work, who they work for, and why. Whether we're fighting cancer, tackling infectious diseases, calming the immune system, soothing neurons, or strengthening the heart and lungs, biomarkers are what make precision medicine real. So how do scientists actually build a genomics test that doctors can trust in a clinical trial? Grab your safety goggles. Let's engineer a biomarker. Everything begins with what's called a context of use, or COU. This is basically starting with the why. Is this biomarker just for learning and exploration? Or will doctors actually use it to make decisions about a patient's treatment? That single decision determines how strict and rigorous the test needs to be. Choosing the context of use is like picking the difficulty level in a video game. Except in this game, regulators are watching your every move. Next, we have to choose our tech wisely. We need the right genomic tool for the job. Do we need a next-generation sequencing or NGS panel to look at lots of genes at once? Or maybe a DNA methylation assay to check for epigenetic changes? We could use RNA sequencing to measure immune activation or tumor signals. For ultra-sensitive mutation hunting, we might use PCR or ddPCR. And if we're looking for villainous microbes, we'd use pathogen detection. The biology of the disease always decides the tool. If we're searching for tiny bits of circulating tumor DNA, we go with deep NGS. If we need to measure a viral load, PCR is our best friend. Now for the fun part, analytical validation. This is the trial before the trial, where we test the test itself. We need to make sure it's accurate, meaning it hits the true answer every time. It must be precise so it gives the same answer repeatedly. We test its limit of detection, or LOD, to see just how tiny a signal it can find. We check its specificity to make sure it finds the right signal and ignores random background noise. And finally, stability. Does the sample survive shipping and maybe a few coffee breaks? Scientists challenge their own assay like it's a ninja obstacle course, using new batches of reagents, different machines, and even different labs. The goal here is to be fit for purpose, meaning the test is good enough for what the trial needs, not overbuilt just for bragging rights. But the lab bench is only half the story. The computers are the brains behind the operation. This is where bioinformatics comes in. Sophisticated software aligns the DNA sequences, calls the genetic variants, and measures mutation frequency, all while filtering out false positives. Every software version has to be documented. Every threshold has to be justified. In this world, data integrity is sacred. No surprise plot twists are allowed in the final results. Here's a truth bomb. The hardest part of genomics often happens before the test even begins. This is called pre-analytic handling. The type of tube used to collect the sample, the processing times, the number of freeze-thaw cycles, and the shipping conditions all matter immensely. If a blood sample sits on a counter for too long, the DNA can fall apart like a cookie dunked in milk. Standardized operating procedures, or SOPs, are absolutely essential to keep the samples healthy and the data trustworthy. Most mistakes hide here, so getting this part right is non-negotiable. So, we've proved we can measure the biomarker reliably. But now for the big question, does it actually mean anything for the patient? This is clinical validation. Does a specific mutation predict which patient will respond to a new drug? Does a particular viral signature warn us that a patient's symptoms are about to get worse? Success at this stage is what turns a biomarker from a simple lab curiosity into a clinical superpower, allowing doctors to stratify patients and give them the best possible treatment. Finally, we have the guardians of good science, quality, and regulatory. Agencies like the FDA in the US or the EMA in Europe have strict regulations to ensure these tests are reliable enough to be used for real patients. Labs follow rigorous quality systems and official guidelines, and they often seek special accreditation. When a patient's health is on the line, safety comes first, data integrity comes second, and maybe coffee comes third. By carefully building, validating, and responsibly using these genomics assays, clinical trials can discover which therapy works for which person at which moment. That is the very definition of precision medicine. The future of healthcare isn't a one-size-fits-all approach. It looks a lot more like you. Thanks for watching. If you loved strolling through our molecular universe today, hit that like button, subscribe for more, and drop your favorite gene in the comments below. We'll see you in the next one.